So today we are going to discuss the selective reabsorption. Now we are not going to draw the full structure of the nephron. Here we have proximal convoluted tubule. This over here, we know that this is the lumen of the proximal convoluted tubule, right? And around the proximal convoluted tubule, we have blood capillaries, and these blood capillaries are called pericular capillaries. We already know that, right? So what is this? These are peritubular capillaries. Now what I am going to do, again I am going to enlarge this much part. Now we know that this membrane is called as basal epithelium, right? Now I am going to draw the basal epithelium like this. Now, this is the basal epithelium. This side is the lumen of the basal epithelium of the this proximal convoluted tubule, and this side over here, the blood vessels are present. And let's suppose that over here we have what? A blood capillary. Now, blood capillary is 1 cell thick, and proximal convoluted tubule is also what? 1 cell thick. Now I have a question over here. The cells that make the lining of the blood capillary, what are those cells called? What?
Squamous epithelium. Endothelium.
Endothelium. Squamous endothelium. Remember that these capillaries, they are generated from the arteries, right? Or arterioles, right? And arteries or arterioles, what do they have? They have 3 tissues: tunica adventitia, tunica media, and tunica intima. And tunica intima is made up of squamous epithelium. Now here I have another question. What are squamous cells like? Flat cells, right? Now when these cells are flat, it means that the things do not have to cross a long distance if they enter into the cells. If they enter into the cells. Now, these are what? These are the basal epithelium. Now, I'm not drawing these nuclei over here, otherwise every cell has its nucleus. Also remember, I told you guys that this over here, this is called as the base of the basal epithelium, right? And this side is called as apex of the basal epithelium. We have also discussed that these are microvilli and villi, right? Over here there are villi and microvilli, both, right? What do? They increase the surface area. And if you pay attention, on the basal side I made the lines more wavy as well. I did not draw the straight lines to show that there are foldings over here, but not as much as villi and microvilli. Understand? So again, even the basal side will have a bit larger surface area, but not as much as the apex. Not as much as the apex. Another thing, that on the basal side, there are special kind of channel proteins present. The next topic which we will study, inshallah, about the homeostasis of the glucose. In that we'll learn about them in detail. Now, these are the special channels that transport glucose. Now, these glucose transport channels, they are called as GLUTs, glucose transport protein channels, right? There are 4 types of these: GLUT1, GLUT2, GLUT3, GLUT4. The number GLUT1, 2, 3, 4 is given according to their location, right? GLUT1 channels, they are usually present in the brain, right? In blood-brain barrier, and the other things we'll discuss later, right? GLUT2s, they are usually present in the liver cells, right? Because liver cells have to take up the glucose to convert that glucose into glycogen. Remember that? I'll ask you this thing as well. And we also discussed that over here, what is present? Core transporters are present. Do you remember this or not? So over here, we have core transporters. And I told you guys that there are 2 types of core transporters. There can be sodium glucose core transporters, and there can be amino acid sodium ion core transporters. Another thing which we discussed, that on the basal side there is sodium potassium pump. Do you guys remember this or not? Right? So what is this? This over here is the sodium potassium pump, right? Now what is happening, that blood is flowing in this direction, right? Over here in the lumen, after the filtrate which is being formed, we are not going into that detail, we have discussed it 2 times, right, already. Now once the filtrate is being made, now over here in the lumen there is filtrate. That filtrate contains what? Glucose. That filtrate also contains amino acids. That filtrate contains urea. That filtrate contains sodium ions. That filtrate contains chloride ions. That filtrate contains calcium ions, right? And etc. Many other things are there. Lactate or lactic acid is also there, right? Because you see, it is the blood which is flowing over here. Now, if you guys remember, I told you guys we do, and there is water as well, obviously. Now, if you guys remember, when I gave you a general lecture about the reabsorption, I told you that in the proximal convoluted tubule there is reabsorption of all the glucose, 100%, all the amino acids, 100%. I told you we don't talk about urea, right? Some of the salts, right? And some of the water, not all the water, not all the salts and minerals, remember? But glucose and amino acids, 100% they are reabsorbed. Now, today it's going to be very easy to understand. It starts from here, from the sodium potassium pump, right? What happens at the sodium potassium pump? It pumps 3 sodium ions into the blood plasma. 3. It pumps 3 sodium ions into the blood plasma and 2 potassium ions from the blood plasma into the cytoplasm of the basal epithelium. Understand? Again listen, what happens? This sodium potassium pump, it will pump 3 sodium ions from the cytoplasm of the basal epithelium into the blood plasma, right? And 2 potassium ions from the blood plasma into the basal epithelium. And during this, what will happen? It will use what? ATP. Right? Now, because of that, what will happen? Because of that, as the sodium is being pumped out over here inside the basal epithelium, the sodium ion concentration will decrease. It is an active transport, right? From lower concentration to higher concentration, sodium is being pumped. Because of that, the sodium ion concentration will decrease where? Over here in all the basal epithelial cells. Let me mention over here, let me show with the green color so that we can see it obviously, right? In all the basal epithelium cells, the sodium concentration has decreased because everywhere what is happening? 3 sodium ions being pumped out like this. 3 sodium ions coming out and 2 potassium ions going in. Because of that, what will happen? Over here, in the lumen, the sodium ion concentration will be what? High. Right? Because the sodium is being pumped from lower concentration to higher concentration from the cytoplasm of the basal epithelium into the blood plasma. Because of that, inside the basal epithelium, sodium concentration is now low, but the sodium concentration is higher in there in the lumen. Now, what will happen? That these are the co-transporters. Through these co-transporters, what will happen? Sodium ions, they will start going in from higher concentration to lower concentration. But as these are co-transporters, what will happen? I am writing only the G, glucose, right? It will tag along with the sodium and it will enter from lower concentration to the higher concentration. From the lower concentration to the higher concentration. Now, glucose concentration is low over here. Where? In the lumen. It is high inside the cytoplasm of the basal epithelium. Now, what is happening? Sodium is moving by simple diffusion from higher concentration to lower concentration. And glucose is just tagging along, right? And will the sodium use energy for moving from higher concentration to low concentration? No. It is, you see, listen, leave that, yes, what I said yesterday, right? Sodium is moving from higher concentration to lower concentration. It is moving from higher concentration to low, so there is no use of energy. As there is no use of energy and glucose is just tagging along with it, so glucose movement is also not using any energy. And this process where co-transportation is taking place of glucose, this is actually called as secondary active transport. Now please understand this concept. Yes. Please understand this concept of secondary active transport. Basically, active transport happened where? Over here. Which generated a gradient, right? That you see, the sodium became higher in concentration over here and low over here. And it happened because of this primary active transport, right? And because of that, what happened? Sodium moved from higher concentration from the lumen to the lower concentration into the cytoplasm of the basal epithelium. And with that, glucose moved from low concentration to high concentration. Now, glucose is moving, just a minute. Glucose is moving from low concentration to high concentration without the use of energy. Without the use of energy. So, we can't call it active transport. We cannot call it active transport. We call it secondary active transport because this potential is generated, this gradient is generated by this active transport. Alright? So what has happened? Now the glucose entered where? Here. into the cytoplasm of the basal epithelium. Now, this glucose is obviously, see, here concentration was low, here concentration is what? High. So from here, the glucose will simply go through these glutes from higher concentration to lower concentration in the blood plasma without the use of any energy. Understand? So glucose is now transported from higher concentration to lower concentration. Same thing will happen for what? For amino acids. Again, this is sodium ion amino acid co-transporter. Sodium ions, they are higher in concentration over here, lower in concentration over there, right? So sodium ions, they will move from high to low concentration. During this process, what will happen? The amino acids, I am writing AA, they will tag along, and amino acids, they will come where? Into the cytoplasm of the basal epithelium. And when they will come into the cytoplasm of the basal epithelium, then these amino acids, through their channels, they will enter where? Into the blood plasma. From higher concentration to lower concentration. Again, remember that in the lumen, the concentration of amino acids is what? Low. In the cytoplasm, the concentration of amino acid is what? High. So what is happening? This gradient of the sodium is generated by what? This primary active transport, right? And the movement of the sodium ions and this amino acid with the sodium without consumption of energy but passing through special channels of the proteins, co-transporter, right? That will be referred as secondary active transport. Understood? Yes.
So in the first place, why does in the lumen, shouldn't the glucose concentration be more?
In the beginning it will be, you see, remember, glucose concentration is lower than this compared, because glucose is obviously absorbed for its own, but for use of energy as well. Right? Remember that over here we did not discuss the internal organelles of the basal epithelium. There is nucleus, I did not know. There are mitochondria as well. Right? Here we are only discussing the parts of the membrane and parts of the cells responsible for transport. Obviously there will be some glutes over here for absorbing the glucose. For which purpose? For respiration. Right? But on the basal side, on the base side, they are extensive in number because their only job is what? Transport. Understand? So here, once the glucose will be absorbed, obviously it will be taken up for what? For respiration, for getting energy. Right? But then what will happen that the glucose concentration and amino acid concentration will become higher over here. Right? So what will happen that these sodium and these, you see, glucose, they will move together. That will be the secondary active transport. Understood? So this is how what happens, that the glucose and sodium and the amino acid, they are transported from the lumen into the blood. Right? So all the amino acids, and all the glucose goes back. 100%. Right? Now understand one more thing. I also said that some of the water also goes back. Now some of the sodium is going back, we have just learned that. Going in from higher concentration to lower concentration. So we have discussed the reabsorption of what? Glucose. We have discussed the reabsorption of amino acids. We have discussed the reabsorption of minerals or you see ions. Now let's discuss the reabsorption of water. Now one thing is very, very obvious over here, that when you see this glucose and amino acid, they will come over here, and I'm saying that glucose and amino acid are higher in concentration over here. So what that will generate? That will generate a higher water potential on the lumen side. Is it obvious or not? Let me draw a simple diagram, another diagram over here of one basal epithelial cell. I am saying that glucose concentration is high over here. I am saying that amino acid concentration is high over here. As I am saying the concentration of these things is higher, It obviously means that over here the water potential will be what? Low. Do you guys remember that we represent water potential by this sign? What is it called? Psi. Right? And it obviously means that the water potential over here will be what? High. So what will happen? That the water will move from higher water potential to lower water potential, passing across the partially permeable membrane. What will be this process? Osmosis. Understand? So then what will happen? This water from here will go where? Into the blood. Understood, everyone? So this is how what happens, that the water is reabsorbed. But remember, yes.
It will pass to aqueduct, right? Not the
Sorry?
membrane. It won't go through the membrane though. The same membrane is.
No, it's entering into the membrane cell. Remember, there is no gap over here. Cells are totally joined.
Entering the cell needs to go through the cell membrane. So it won't be able to go through the membrane itself.
No. Obviously, we know that.
Go through
Aquaporins. Yes, right? That's why we are saying partially permeable membrane, right? And it passes through aquaporins. When I am saying, remember, when I am saying passing through cell membrane, it obviously means aquaporins, because we already have this knowledge, right? That's why I did not go into that detail. But we will talk about aquaporins, inshallah, when we will talk about the reabsorption of the water in distal convoluted tubule. Alright? Because there the role of aquaporins is a lot. Everyone understood this? Yes.
So why aren't all the ions reabsorbed if there's always a concentration gradient?
Or because you see, the filtrate is moving. It's not stagnant at one place, right? As it is moving along, so obviously rest of the amino acids and rest of the sodium ions, chloride ions, calcium ions, oxalate ions, they will move forward. Right? Not even the whole water is reabsorbed. Right? Rest now you see next thing which we are going to discuss, that is the reabsorption of water and minerals now. Water and salts now. Alright? Everyone understood this? Now quick overview just in 1 minute. First thing, active transport on the basal side. Please pay attention everyone. Right? Active transport on basal side. 3 sodium went outside, 2 potassium came inside. So sodium ion concentration became low over here and higher over here in the lumen side. So the sodium ion started moving from higher concentration to lower concentration. Tagging along is what? Glucose. From lower concentration to higher concentration. Now glucose is moving from low to high concentration without the use of energy, taking the help from sodium, which is passively diffusing. Right? So that's why what is it called? Secondary active transport. There can be a question, define what is secondary active transport. Right? Answer is very, very simple, that the glucose or amino acids, they are tagging along and using the gradient of the sodium to move from lower concentration to higher concentration. That is called secondary active transport. Same way what will happen, amino acids will also move. Then with the help of special channels which are called glutes, I mentioned over here, right? The glucose will move from higher concentration in the cytoplasm into the blood plasma. And same the amino acids, they will move through their own channels from the higher concentration to the lower concentration into the blood plasma. Water will move from higher water potential to lower water potential. And this potential difference is caused by what? By the higher concentration of glucose and amino acids. Now you understand? So this is how selective reabsorption. Now remember, I use the word selective reabsorption. So I have talked about the reabsorption of glucose, of amino acids, some sodium and some amount of water. And we did not talk about urea. Now remember, I told you in my last lectures, urea is also reabsorbed. That is, but at the end, this is the net movement. That is the net movement. Urea will be reabsorbed if there is concentration is higher, where? In the blood plasma. Understand? But at the end, it is the net movement. How much net urea is being removed out of the body. Alright? Now we are going to discuss the reabsorption of water and salts. For that purpose, I am not again going to draw the whole of the nephron. I am just drawing the loop of Henle. Now everyone, we know that this is loop of Henle and this part is called as descending loop of Henle and this part is called as ascending loop of Henle. Now we know that why do we call it as descending and why do we call it as ascending? That is because of the movement of the filtrate. In the descending loop of Henle, the filtrate is moving in downward direction, and in ascending loop of Henle, the filtrate is moving in upward direction, right? We also know that around the loop of Henle, there are blood capillaries, and those blood capillaries, they are called what? Vasa recta. Now very quickly, now these are Vasa recta, now if you guys remember, I have taught you that the vertical and horizontal, these capillaries, these are called what? Vasa recta. And these gaps over here, these are called what? Interstitial spaces. Right? Now, what will happen? When the filtrate will come down, first reabsorption of salts will start. First, the reabsorption of what? Salts will start. What will happen from here? Sodium ions and chloride ions, they will be reabsorbed where? Into the interstitial spaces. They are not going directly back into the blood. They are not going directly back into the blood. When the sodium ions and chloride ions, they will be reabsorbed into the interstitial spaces. Because of that, what will happen? The sodium ion concentration, chloride ions concentration, it will become what? Higher where? In the interstitial spaces. Because of the higher concentration of these solutes, because of the higher concentration of these solutes, what will happen? Over here, the water potential, I am writing W, will become what? Low. The water potential will become what? Low. Now what will happen? The water will start moving from higher water potential to what? Lower water potential. Water will start coming from the descending loop of Henle into the interstitial spaces. Again, water is not directly going into the blood. Water is first coming where? Into interstitial spaces. From higher water potential to lower water potential. Then from the interstitial spaces, the water will go where? Into blood. Water will go where? Into the blood. And this is how the water is reabsorbed. Now here, one thing which we need to discuss, that is the counter current multiplier. Now, over here the filtrate is going downward. Over here the filtrate is going upward. Filtrate is moving in two opposite directions, right? So we will call these the movement of this filtrate in two opposite direction as counter current. What we'll call it as counter current. Counter means opposite. And multiply means what? If more salts will be absorbed, then there will be more reabsorption of what? Water. Increase in reabsorption of salts will increase the reabsorption of water. Right? So that's why it is called what? Counter current multiplier. It is just like directly proportional. More reabsorption of salts will lead to more reabsorption of water. Means that this effect will multiply the effect of this. That's why it is called counter current multiplier. Another very important thing which we discussed yesterday as well, that over here in the bottom of the loop of Henle, the concentration of filtrate will be highest higher than the descending loop of Henle, higher than the ascending loop of Henle. How? Now over here, when the filtrate is coming down into the descending loop of Henle, it contains a lot of water, right? So obviously it will be dilute. Here, when it is going up in the ascending loop of Henle, it is losing salts. It is losing sodium and chloride ions. It is losing solutes, right? So obviously, as it is losing solutes, the concentration will be low over here as well. But over here at the bottom, both of these things are not happening, right? Right at the bottom, right in the middle of it. So that is the reason the over here, in the bottom over here, the concentration of the filtrate will be what? Highest. Understood? This is again, over here, a lot of water coming down. Over here, it is losing sodium. Obviously, there is potential difference over here, right? But still, the water will over here will be more than this, because here it is losing a lot of solutes. Got it? So this is how what happens, reabsorption of salts and water take place. But remember one thing, this question should have come in your mind. In the beginning, obviously sodium will go outside and chloride ions, they will go outside by diffusion. But if there is need of more or extra reabsorption of water, because you guys did not drink enough water and you did sweat a lot, then what will happen? No, ADH role will come later. Then here, the reabsorption of these salts can also take place by active transport. Here can be what? Sodium, potassium pumps are also present over here, right? Which will pump 3 sodiums outside, 2 potassiums inside. Understand? That will also help in producing what? A gradient, right? That will also help in reabsorption. Understood, everyone? Alright.